Hello students, we meet here today for yet another session for the NPTEL certified online course on the topic An Overview on Maternal Health, the Antenatal, Intranatal and Postnatal Care. I am Dr. Barnali Ghosh, an obstetrician and gynecologist working at Viceroy Medical College and Medical Research Center, IIT Kharagpur. In the last classes, we have already discussed regarding the anatomy of female external and internal genitalia, an overview of the anatomy of the pelvic floor and perineal muscles. Today, we are going to discuss on gametogenesis. Obstetrics as a whole, it deals with pregnancy and pregnant female. This pregnancy is the result of fertilization of two gametes, that is the male and the female haploid gamete, which ultimately forms the diploid zygote or the embryo which forms the fetus. So, today, the process of formation of these gametes, that is gametogenesis, is our topic of discussion. Concepts covered in today's class: spermatogenesis, oogenesis. The key words being spermatogenesis, oogenesis, sperm, ovum. So, here this is a pictorial representation of fertilization, which is the union of two haploid gametes, the male gamete known as the sperm and the female gamete which is called the ovum. And the process of formation of this sperm is called as spermatogenesis, and oogenesis results in the formation of ovum. The union of these two will result in the formation of zygote, which is diploid. These two are haploid, ultimately forming a diploid zygote. To note, gametogenesis starts from primordial germ cell. This primordial germ cell or primitive germ cell, embryologically they develop from the epiblast, right? So, they develop from the epiblast and they travel to the dorsal surface of the yolk sac at 4th week of intrauterine life. So, at 4th week they travel to the dorsal surface of the yolk sac. These are primordial germ cells. Then it will then reach the genital ridge at 5th week of intrauterine life, right? So, these genital ridge, we know they ultimately result in the development of ovary and testis, that is the gonads, depending on the presence of Y chromosome. Presence of Y chromosome, dictates the genital ridge to form into the testis, and in absence of Y chromosome, by default ovary is produced. So, these primordial germ cells, they get embedded or they are present within these developing gonads, and they form spermatogonia in males, oogonia in females. Another talk on teratoma: if from these primitive germ cells, if they deviate from their usual path and if they reach, sometimes they reach the neck region. If they reach the neck region, then these primitive germ cells will result in the formation of oropharyngeal teratoma, right? These are actually formed from the primordial germ cell, which instead of going to the genital ridge, which is its usual position, they get deviated and may reach the neck region, leading to formation of teratoma. They are actually pluripotent cells. What that means? It means that these are capable of formation of all the three layers, that is the ectoderm, the endoderm and the mesoderm. So, tumor originating from these primordial germ cells will result in formation of teratoma. Sometimes they may get they may deviate and reach the sacrococcygeal region, and there ultimately they form sacrococcygeal teratoma, right. So, this is part of the embryology development, and now we know that the primordial germ cells, which actually form the ovum or the sperm, they actually develop from the epiblast or the dorsal surface of the yolk sac. Coming to the concept of haploid, diploid or triploid, this is a haploid cell. So, what that means, that the chromosome, this is one chromatid, right. Maybe this, I will say this is the chromosome number say 9. This has one chromatid. Here this is the same chromosome containing the same genetic material, but it has 2 chromatids. Here there are 3 chromatids. So, depending upon the number of chromatids, it is haploid containing only single one chromatid. When 2 chromatids are present, it is diploid cell, and each chromosome has 2 chromatids. So, or if it is 3, then it is triploid. So, the gametes are actually haploid cells. Rest all the cells of all the body organs, they are actually diploid cells. What is the chromosome number? In case of humans, it is 46. In females, it is XX. In males, it is XY, right. And in gametes, it is haploid, so 23X or 23Y. We will go into the topic. So, what are the types of cell division? Cell division, number 1 is mitosis and 2 is meiosis. Mitosis is just replication of one cell to 2 daughter cell. There is no decrease or increase in the nuclear material. A haploid cell, or here it is a diploid cell, 2 ends. So, a 2N cell when undergoes mitosis will form 2 2N cells, right. But in meiosis is a reduction division. In meiosis, reduction division. So, what happens in meiosis, 1 2N or diploid cell, this is the parent cell. Here it will form 4 daughter cells, but they are haploid, right. So, here each of the cells have less chromatin or less nuclear material. The daughter cells are haploid. From diploid cells, they are forming haploid cells. So, this is a reduction division. This meiosis occurs in the gametes or the gonads. Gametes are present in the gonads. So, meiosis occurs only in the gonads, mitosis occurs everywhere, in the brain, in the skin, in the liver, any other organ of the body. Mitosis occurs, but in gonads, meiosis occurs for the production of gametes. Coming to meiosis proper, meiosis now can be divided into meiosis 1 and meiosis 2. This is actually mitosis, 2N forming 2N cells, so it is mitosis. Now, this 2N will undergo meiosis division, meiosis 1, where there will be reduction in the nuclear material forming 2 haploid cells or containing n number of chromosomes. So, coming to meiosis II. Meiosis II is nothing but similar to mitosis. Here one cell containing n number of chromosome will form 2 cells that will contain the similar amount of nuclear material, that is n number of chromosomes, right. So, from a diploid cell we are getting 4 haploid cells by the process of meiosis. Meiosis I, meiosis II. Each stage of meiosis next can be divided into 4 phases. They are the prophase, metaphase, anaphase and telophase. And this prophase phase can again be divided into 5 stages. What are they? Leptotin, zygotin, pachytin, diplotin and diakinesis. So, these are the stages of meiosis, meiosis I, meiosis II and the 4 phases of meiosis, that is prophase, metaphase, anaphase and telophase. Coming to spermatogenesis proper. So, cells of spermatogenesis. Where does the spermatogenesis occur? It occurs in testes. So, cells of the testis, they are 2 types, seminiferous tubular cells and the interstitial cells. Within the seminiferous tubule, in inner testis, there are approximately 300 to 400 seminiferous tubules, and within each of these seminiferous tubules, there are again 3 types of cells. First is, 2 types of cells. First is the Sertoli cell, and this is also called as the sustentacular cell, and other is the spermatogenic cells or the male germ cells in different stages of development. In the interstitium, there are 3 types of cells: Leydig cells, fibroblast and neurovascular bundle, right. So, this is the structure of the testis, where these are the seminiferous tubules. There are 300 to 400, so many seminiferous tubules are present. So, these are the seminiferous tubules, and they drain or they are joined and they form the rete testis, which ultimately will connect with the epididymis. So, this is the tail of the epididymis, this is the tail of the epididymis and the head of the epididymis. This whole structure is the epididymis, which will ultimately connect to the vas deferens, which goes ultimately forming the ejaculatory duct and which will help in ejaculation, right. So, we will now study each of the seminiferous tubules. Cross section each of these seminiferous tubules, we are studying a single seminiferous tubule. The cross section has been taken. Now, within the seminiferous tubules, there is a lumen at the middle. This is the lumen at the middle, and surrounding the lumen, these are the Sertoli cells. These violet colored structures are the Sertoli cells. These will help in anchoring the male germ cells in different stages of development. This spermatogonia, this is the first male germ cell. Spermatogonia will undergo spermatogenesis in different stages, in different steps of the spermatogenesis, and they are attached to the wall of the sustentacular cells or the Sertoli cells. In the lumen, then these spermatozoa, which are the end product of spermatogenesis, they get dislodged and they will come into the lumen of the seminiferous tubule. Outside this seminiferous tubule is the interstitium, and in the interstitium there are 3 types of cells. These brown colored are the Leydig cells, these are the vascular bundles, and these are the fibroblasts, right. So, we will now study about the Sertoli cells. See, these Sertoli cells, this is a Sertoli cell, right. So, this Sertoli cells and the adjacent Sertoli cells, they are connected by tight junctions, and this forms the blood testis barrier. There is a tight junctions, and any material like in the blood vessels, there can be toxins, there can be chemicals. So, any material cannot pass through this blood brain barrier, it cannot pass. This is called as the blood testis barrier, which is formed by the Sertoli cells, and this helps to safeguard the gametes of the testis, like the male gametes. It will safeguard the gametes from any external circulating carcinogen or chemicals, right. So, one function is formation of the blood testis barrier. Next is these Sertoli cells, as they help or anchor the male gametes, they will provide nourishment to these gametes, right, and also it has a hormonal activity. It secretes androgen binding protein, it secretes inhibin B, and it secretes the transferrin. These are the hormones secreted from the Sertoli cells, and these cells are under the action of FSH. The Leydig cells are outside the blood testis barrier in the interstitium, and they secrete testosterone, right. They secrete testosterone, and they are under the action of LH. So, we have discussed these are the Leydig cells which will secrete the testosterone, and these are the single are the Sertoli cells. They are present within the seminiferous tubules, right, and these are blue colored, these are the male germ cells in different stages of development, and this is the lumen which will contain the spermatozoa, right. So, first the tail gets dislodged, then the head of the spermatozoa into the lumen. Sertoli cells, yes, what are the functions of Sertoli cells or sustentacular cells? They provide nourishment to the male germ cells. They form the blood testis barrier by the tight junctions, and thereby safeguarding the germ cells from the external any insult, and there is also secretion of hormones from these cells, releasing of androgen binding protein, transferrin and inhibin B. Another point here is this androgen binding protein, these will help to concentrate the testosterone. Testosterone is released from the Leydig cells, and this testosterone will gain entry into the seminiferous tubules, and this androgen binding protein which are released from the seminiferous tubule, they will help to concentrate that testosterone within the seminiferous tubules. The testosterone concentration is as high as 50 to 60 times inside the seminiferous tubules in comparison to seminiferous tubules, in comparison to the plasma concentration. So, this increased concentration of testosterone within the seminiferous tubule is required for the process of spermatogenesis, right. So, Sertoli cells we have discussed. Now, the spermatogenic cells or the male germ cells, they actually start from spermatogonia. Spermatogonia will develop from the primordial germ cells. We have talked about it. It arises from the arises in the yolk sac from the epiblast, and they come to the genital reach, and they enter the testis by 5th week of intrauterine life. Leydig cells, they are outside the seminiferous tubules in the interstitium. They secrete testosterone. They are under the influence of LH, and they are present outside the blood-brain barrier, right. And because it secretes testosterone, it helps in the formation of secondary sexual characters in male. Now this is the HPO axis. What happens? The HPO axis is activated at puberty. Spermatogenesis starts from puberty and it continues till death. Even we can collect spermatogonia or spermatozoa or the sperm can be collected from the testis of a dead man, right, even 48 hours after his death. So, this spermatogenesis starts from the puberty and it continues throughout his life until death. What happens? There is activation of HPO axis at puberty. This is the hypothalamus, pituitary and ovarian axis. Pituitary, HPO axis is hypothalamus, pituitary and ovarian axis, or in females and testis in case of males. So, hypothalamo-pituitary gonadal axis, right. So, the hypothalamus will be secreting GnRH. This is getting activated at puberty and GnRH is being released from the hypothalamus. It will activate the anterior pituitary, which will secrete FSH and LH. This FSH and LH will activate the testis and there will be production of testosterone, which have negative feedback on the hypothalamus, thereby decreasing the secretion of GnRH, and also thereby there is decreased secretion of FSH and LH. So, this cycle goes on. In females similarly, from the ovaries there is production of estrogen, which will then again have feedback inhibition on the hypothalamus. So, the HPO axis activate at puberty. Spermatogenesis start from puberty and continue till death. Whereas in females, this is a point of differentiation. The oogenesis start in the intrauterine life before birth of the female fetus, but it is arrested in the prophase of meiosis 1, and this meiosis 1 resumes at puberty after the activation of the HPO axis in females. Coming to the steps of spermatogenesis, there are 5 steps. Number 1 is from spermatogonia to primary spermatocyte to secondary spermatocyte, then to spermatids and ultimately spermatozoa or the sperm, right. So, we will come to one by one. This is a 2N cell. This is called as spermatogonia. This is a diploid cell. They are present in the testis. They are formed from the primordial germ cell and are present from birth up to the puberty. At puberty, the spermatogonia and the primary spermatocyte. This is primary spermatocyte. This is formed from spermatogonia by the process of mitosis. Then what happens? Now, this is the onset of puberty in males. Then the process of meiosis starts, right. So, meiosis 1 is a division of reduction in nuclear material. The 2N diploid cell will become haploid cell, and these are called as the secondary spermatocyte, right. Ultimately, each of the spermatocyte will undergo each of the secondary spermatocyte will undergo meiosis 2 and they will form 4 spermatid. This spermatid will undergo transformation, in its structure, in its content of cytoplasm, in its tail formation, and they will form ultimately spermatozoa or the sperm. Point to note is from spermatogonia to the production of spermatozoa, this full process is called as spermatogenesis. And the process by which spermatid is develops into the spermatozoa. This is only development or a transformation in the cell, and this process is called as spermiogenesis, right. So, spermiogenesis is not mitosis or meiosis. No change in the nuclear material occurs between the spermatid and spermatozoa. The nuclear material in spermatozoa and spermatid are same, but there is change in the structure of the cell, right. The spermatozoa has tail, and this spermatozoa, when they get dislodged into the lumen of the cell, this is called as spermiation. This process of dislodgement of the spermatozoa from the wall of the sustentacular cell into the lumen of the seminiferous tubule, that process is called as spermiation. The duration of spermatogenesis is 60 to 74 days, of which the duration of this spermiogenesis, this duration is 10 to 14 days. This is a pictorial representation where we can see the Sertoli cells. This is the blood testis barrier. Within this is the seminiferous tubule, and a part of that seminiferous tubule showing the Sertoli cells and the different male germ cells in different stages of development. And this is the lumen where the spermatozoa would get dislodged from the wall of that sustentacular cells. They will go into the lumen, and from the lumen it will be transported through the ducts. Duration, yes, 60 to 74 days. Location of spermatogenesis is in seminiferous tubules. It is in seminiferous tubules. From the seminiferous tubule, from the lumen, the spermatozoa will go into the epididymis, right, and in the epididymis there will be maturation as well as attainment or maturation and motility of the sperm. This occurs in the epididymis. What is the purpose of spermatogenesis? It is to form sperm or male gametes. Now, coming to structure of sperm, it is a single cell. It has a nucleus which forms the head of the sperm. The Golgi apparatus, they form the acrosomal cap. Mitochondria forms the middle piece. The centrioles, they form the neck of the sperm, and axial filaments in arrangement of 9 plus 2, it forms the tail of the sperm. There is a specialized protein called the dynein protein in the tail which helps to assimilate these microtubules. So, this is the sperm and this is the acrosomal cap. This is the acrosomal cap, and this contains Golgi apparatus. This is the head of the sperm which will contain the nucleus, haploid nucleus, right. So, this part is the neck containing the centrioles. This is the middle piece which contains the mitochondria which gives energy to the sperm for its motility, and this is the tail of the sperm, right. Coming to the length, yes, it is from this approximately length comes out to be 60 micrometer, length of a single spermatozoa. So, this is the spermatogenesis. Coming to oogenesis. Oogenesis in females, it starts in the ovary, right, the female gonads, and it starts in intrauterine life before birth. At 20 weeks of intrauterine life, there is 7 million oogonia, which comes down to 2 million at birth, right. So, and then at puberty, the number of primary oocytes is 4 lakh in both the ovaries. Then during the reproductive years, during the reproductive age group, which is approximately 15 to 45 years of age, the ovulation, there is ovulation of approximately 400 to 500 secondary oocytes, and at menopause there is less than 1000 oocytes in the ovaries. So, there is gradual decrease in the number of oogonia or primary oocytes in the ovaries with age. As the age increases, the number of oocytes decreases and thus decrease the ovarian reserve. So, oogenesis proper, same as spermatogenesis, it starts from oogonium, which is a diploid cell. It forms primary oocyte and this is also diploid. This primary oocyte will undergo meiosis 1 to form, here is the difference, it will form one secondary oocyte and one primary polar body, right. So, secondary oocyte in case of males, secondary spermatocyte was 2 from a single spermatogonia, but in females one secondary oocyte is being produced from one single oogonium. Secondary oocyte will undergo meiosis 2 to form one ovum and this is a polar body, right. And these two are also polar bodies. The 3 polar bodies are formed. From oogenesis, only one ovum is being produced. From spermatogenesis, 4 sperms are being produced from a single cell of spermatogonia, right. Now, we have known, yes, in the fetal ovary, in the fetus before birth, in the intrauterine life, these female germ cells, they are present as oogonium, which is diploid 44XX. They form primary oocyte in the fetal life, in the fetal ovary, and these primary oocytes, they get arrested. This meiosis 1 will get arrested at the prophase, diplotin stage of prophase of meiosis 1. Here the meiosis 1 gets arrested and this arrest remains till puberty. After puberty, with the onset of puberty, with the activation of the HPO axis, this arrest goes off and meiosis 1 is completed, thereby producing the secondary oocyte and one polar body. So, this is at puberty, right. So, first phase of meiotic division is completed at puberty just prior to ovulation and it enters into the second meiotic division, that is meiosis 2. Secondary oocyte now entering the meiosis 2 and again it gets arrested in the metaphase stage. This metaphase stage arrest will go away only if the ovum or this secondary oocyte is fertilized by a sperm. If there is no fertilization, then the secondary oocyte will not complete the second meiotic division and it will get destroyed. Coming to the ovarian follicles, these female germ cells are they get they are contained within the different ovarian follicles which are present in the ovarian cortex. So, till puberty, the female germ cells remains as primary oocyte. So, this is the primary oocyte, this is the primary oocyte, right. This is the primary oocyte and primary oocyte gets covered by granulous cells. This forms the primordial follicle. Slowly along the stage of development, they will form primary follicle, then the secondary follicle, and this is the mature tertiary follicle, right. So, this is also called as the antral follicle, which will ultimately form the Graafian follicle at the time of ovulation. This is the mature Graafian follicle which will undergo ovulation and thereby release of secondary oocyte. Up till this is the primary oocyte, but just prior to ovulation, the primary oocyte gets converted to secondary oocyte, which is released into the peritoneum during the process of ovulation, and the rest part of the ovarian follicle will get converted to corpus luteum, and if there is no pregnancy, this slowly gets atretic to form corpus albicans. So, oogonia form primary oocyte. Just a revision, primary oocyte remains as primary follicle in the intrauterine life. They are arrested in the prophase, that is the diploting stage of prophase of meiosis I. At puberty, after the establishment of HPO axis, there is release of FSH and LH, right. So, this is a primary oocyte. This is 2N. We cannot say 2N, but it is arrested, right. It has not been complete, the first meiotic division has not been complete. So, it is a primary oocyte, and these are the cuboidal, flattened cuboidal cells all around. This structure is called as the primordial follicle, which is present in the fetal ovary. Now, this primordial follicle will form the primary follicle where the flattened cells will become cuboidal, right. But inside is the same primary oocyte. With puberty, with the action of FSH, there will be growth of, these are the cuboidal cells, they are nothing but the granulosa cells of the ovary. There will be growth of the granulosa cells and it will form the secondary follicle, right. Now, and these are the theca cells. This is the secondary follicle, and this also contains the primary oocyte, right. Now, slowly there will be fluid cavity inside the granulosa cell. This is the fluid cavity, this is called as the antrum. With the appearance of antrum, the secondary follicle is called as the tertiary follicle or the antral follicle. So, granulosa cells around the primary oocyte leads to secondary follicle, and this secondary follicle, within the secondary follicle there is a formation of cavity or antrum which leads to the formation of tertiary follicle. This tertiary follicle, there are many tertiary follicle, and from there only one dominant follicle is selected, and that dominant follicle will form the Graafian follicle, which is approximate size is 18 to 20 millimeter. So, that this Graafian follicle with LH surge, till then this is the primary oocyte. After the LH surge, this primary oocyte, there will be resumption of meiosis I, the arrest will go away and it will form the secondary oocyte and the first polar body. This secondary oocyte is, this is the secondary oocyte which is getting dislodged, or it will get rupture into the peritoneal cavity during the process of ovulation. So, this is as a whole primordial follicles converting to primary follicle, secondary follicle, tertiary follicle, all containing primary oocyte arrested at prophase of meiosis I. Then the Graafian follicle or the mature follicle just prior to ovulation, and from the Graafian follicle during the process of ovulation there is release of the secondary oocyte. Now, to sum up, from the follicles in the ovary, from the follicular pool in the ovary, some follicles are they are selected or they are recruited from that follicular pool and they go into the process of oogenesis, right. So, this recruitment of follicles from the pool, this is not FSH dependent. Up till the formation of preantral, up till the formation of secondary follicle. From the preantral it is FSH, before preantral it is gonadotropin independent. Then from preantral they become FSH dependent, and gradually FSH causes proliferation of the granulosa cell, and there will be formation of preantral, antral follicle, ultimately Graafian follicle or the dominant follicle, which under the action of LH will rupture causing ovulation, right, and the rest of the follicle there will be luteinization to form the corpus luteum. This is the same, we are slowly towards the right side, we are going into the more advanced stages of ovarian follicle. Another thing to note is when the during ovulation what is released. Number one is the secondary oocyte. This is released, this is the secondary oocyte, and around the oocyte is the zona pellucida, a membrane. This is the zona pellucida, this red portion is the zona pellucida, and a part of the granulosa cells surrounding the oocyte. These are arranged in a form of rays. This is known as corona radiata. They are nothing but the granulosa cells surrounding the oocytes. So, this is released during the process of ovulation. Right. So, this is oogenesis. To summarize, oogenesis, from oogenesis a primary oocyte will ultimately form a single ovum, right, and 3 polar bodies. This is the first polar body and these are the 2 polar bodies. So, single ovum is produced from one primary oocyte, but in spermatogenesis one primary spermatocyte will form 4 sperms, and the fertilization between this ovum and the sperm will result in the formation of zygote. Now, coming to some of the MCQs, right. MCQ তো আসেনি? গন্ডগোল হয়ে গেছে।
হ্যাঁ, রেফারেন্সটা আগে
এখান থেকে বলি নাকি, না এমসিকিউ থেকে বলি? বলবো?
হ্যাঁ ম্যাম, ওটা ফুল স্লাইড করে দিন না।
Now, coming to some of the MCQs in this chapter, oogenesis starts in the early fetal life. The basic difference between spermatogenesis and oogenesis is mature ovum is diploid and sperm is haploid. No, one mature ovum is produced in oogenesis and 4 mature sperm is produced in spermatogenesis. Yes, one mature ovum from one oogenesis. Right. So, this is the correct statement, rest are all incorrect, you can read through them. Acrosome of sperm is formed from acrosome, it contains the Golgi apparatus. The process of spermatogenesis is induced by it is induced by FSH. Actual genetics section of sperm is its, that means where is the actual gene, that is the nucleus of the sperm, it is in the head of the sperm. Polar bodies are formed during, yes, in oogenesis. The surge in LH that occurs during the middle of ovarian cycle triggers LH surge will trigger in ovulation, right? LH surge occurs just prior to ovulation, which will result in ovulation. Which type of cell is actually ovulated from the ovary? From the ovary, the cell is secondary oocyte. Primary oocyte is arrested from the intrauterine life and it remains in that arrested position. Just prior to ovulation, this primary oocyte, the arrest goes away and it forms secondary oocyte. So, during ovulation, secondary oocyte is released. Second polar body is released from the oocyte. Second polar body, that means in meiosis II, the secondary oocyte will undergo meiosis II to form the ovum and the second polar body, and this meiosis II arrest goes away only after fertilization. So, answer D, few hours after the sperm enters the oocyte, that is after fertilization. Ovulation coincides with LH surge. LH surge occurs 12 to 14 hours before ovulation, right. So, this is the end of this session. Reference taken are from DC Datta textbook of obstetrics, the Gray's Anatomy and the Williams obstetrics. Thank you.